Well, good afternoon, everybody. My name is Dr. Brian Bader. I'm an entomologist with the University of Florida, and I specialize in ornamental insect vector ecology. And by trade, I'm an entomologist, so I've kind of gotten all my degrees, and my early research was all based on insects. But during my PhD and postdoc, I started getting trained in plant pathology. And with my current position, it's kind of bringing those two fields together where I deal with pathogens that affect plants, but also the insects that transmits them. And I was hired at this position in 2016, specifically to look into the disease lethal bronzing. Now, when I first got here and early on, the disease was called Texas Phoenix palm decline, but we kind of rebranded it to be a little more um, scientifically accurate and representative of insects. But during my PhD, symptoms and also to try and um, remove the stigma which comes along with putting a place name in the title of a disease, specifically because we're seeking SCRI funding from the USDA, and they need a multi-state component. And I know the Texans didn't appreciate their name on the disease. So um, both for scientific and political purposes, we decided to rename it lethal bronzing, and it kind of fit the scheme of uh, lethal yellowing, which is another phytoplasma disease that historically has been in Florida and killed off quite a bit of coconut palms. So today I'm going to present kind of the current state of the disease here in Florida. I'm going to give a little bit of background on lethal bronzing, and then I'm going to shift into more focus on where we're at with managing the disease and specifically with regard to vector control. So, as I mentioned, the disease was called Texas Phoenix palm decline, and that is because it was initially described in Texas in 1980. Now, that was before we had access to molecular techniques that are used to determine what pathogens are involved with different diseases. So it wasn't until about 2002 that they characterized at the molecular level with the pathogen that had been affecting Texas. So I guess from 1980 to 2002, they suspected it was lethal yellowing because lethal yellowing was in the area. But in 2002, they determined it to be a different strain of phytoplasma, which belongs to what is called the palm lethal decline group. And they used a gene called the 16S gene for the taxonomy. So there's lots of different types of phytoplasmas in the world, and they're named 16SR Roman numeral 1 through, I think we're up to like 40 at this point. And they're just numbered that based on the sequence of discovery. Then within each of those groups, you have strains or subgroups. So the palm lethal decline group is group 4, and we have subgroups A, B, C, D, E, and F. Lethal yellowing is caused by subgroup A or strain A, and lethal bronzing is caused by strain D. Now, the other strains are kind of obscure ones that are kind of sporadic throughout the Caribbean. And C actually probably doesn't even belong in the group 4 classification. It's from Africa and and while it causes the similar symptoms at the molecular level, it's very distinct from these other ones in the Caribbean. So subgroup D is what was found in Texas in 2002. Then in 2006, they had some declining palms in Hillsborough County and on the west coast of Florida. And when they tested the palms, they found that they were testing positive for phytoplasma, and when they did the follow-up analysis, it turned out to be the same strain that was in Texas. And it was in this map on the slides here, you can see in 2008, those 3 counties are where it was first documented from. And from 2008 to 2018, you can see that it has spread throughout much of the state. I need to update that map. We have a few new county records. The one that would be most evident on this map is that last one there on the Panhandle. I think it's Escambia County. I have it here in the table, where I guess that's where Pensacola is. But we've had a positive from there, which is not terribly surprising because the disease has been documented in Louisiana also. So that whole southern, the southern part of the states that are bordering the Gulf Coast there on the north are probably all suspect. Not to mention Texas has it. So I think that whole arc going across the Gulf is at risk also. Now you can see as far north, it goes all the way up to Jacksonville. And I'm starting to get emails from people in Georgia with suspicious looking pictures of cabbage palms specifically. And it ranges all the way, the southernmost record in Florida is Key Largo. But to this day, we still see the highest number of infections occurring in the central part of the state. Basically, excuse me, from Tampa area going kind of southeast towards Fort Pierce, that's where you see the majority of infections. Now, since I started the job here when I got here in 2016 and we were driving north on the turnpike, where you really started to notice a lot of infections was the Fort Pierce area, where I think it's State Road 70 that is the interchange between the turnpike and 95, and that's where you really see a lot of infections. Then over the years, I've seen it creeping further south to now where if you're driving north, you start to see a lot in West Palm Beach. So it's on the move south, I think faster than it is north. And I suspect that is due to the just abundance and density of susceptible hosts. For the most part, once you get north of Orlando, at least from what I can tell, most of the palms you come across are wild cabbage palms growing along the roads or in natural areas. And because of this, it's harder for the disease to really take effect and spread rapidly. Whereas you go further south, you have a lot more density of palms, a higher density of palms, and greater diversity. So we're starting to see it jump into new host species as well. But I think because there's fewer palms and they're farther between the further north you go, that's why the disease isn't as evident further north. That's not saying it's not there. I know in Gainesville, they very wisely planted a bunch of Phoenix palms, knowing full well that they're highly susceptible, and it's starting to flare up a lot more in Gainesville. And I'm getting more and more samples from Jacksonville area as they're planting all of these Phoenix palms. Now, the interesting phenomena is that the majority of the spread is likely occurring in the nurseries, where these Phoenix palms are kind of planted in a monoculture, and you have a high density of susceptible hosts, so it's rather conducive to disease spread. And basically what happens is, because of there being a lot of weedy habitat and grass, you have high densities of the vector. And what happens is you get huge amounts of spread occurring in the nursery. And then From time to time, infected palms get shipped out. And usually what happens is when these infected palms get planted in a new spot in an urban setting, like along a major roadway or a business park, that's where the public sees it. That's where it's most notable. So, and we do see secondary outbreaks, but it's less, I think it's less likely from the palm actually allowing further spread by the vector in the area. But what I think is happening is that when these palms are moved from the nursery, there are insects on them already that are carrying the pathogen. So when it's not that the palm is being the source once it's planted, because it dies very quickly. But once it gets brought to the new area, the vector jumps off and causes secondary outbreaks. So that's why we see a lot of cabbage palm in areas where they're either natural but really close to human habitat, or in some cases that are like around the nurseries that are closer to more natural habitats. I've seen a lot of cabbage palms dying in those areas as well. Now, down in Broward at the research center here in Davie, we had an outbreak of the disease here on the station, and it basically went through and has wiped out quite a few of our cabbage palms on the station. So that's just a little bit of info on the dynamics of the disease spread and how it's moving. And I kind of got a little bit ahead of myself on the host plants, but it's the next slide, so it's I think it's fine. This is our current host range that we know from the state of Florida. Now, most of these palms, as far as I'm aware, I think the only native palm on the list of susceptible hosts is Sabal palmetto, cabbage palm. The rest of these palms are ornamentals, and I think maybe the only ones that are maybe potentially naturalized at this point down here are coconut and queen palm. Maybe Robolini, but I don't know. But basically, all of these palms, even cabbage palm, is used as an ornamental. And as I mentioned, as we see the longer this disease goes on, the more new host plants we're getting. But interestingly, not all of the hosts on this list are what I would call equally susceptible. The ones that we get hit the worst are our phoenix palms, Canariensis, Dactylifera, and Sylvestris specifically. From what I can tell, cabbage palm is highly susceptible. We get a lot of infections, and queen palm is kind of moderate. The rest of those host plants, it's not common. It's just a few sporadic ones. Now, I don't know if this is due to true host susceptibility or resistance to the pathogen, or if it's related to the vector feeding preference, or a little bit of both. And I'll touch a little bit on the potential resistance of different species in a little bit with some other data that I have. But basically, the list keeps growing, and the coconut palm is one of our newest additions to the list, and it's one of the concerning. It's concerning because it kind of revokes reignites the concerns and fears that lethal yellowing had on coconut palms back in the 70s, 80s, and 90s. But this is our current list of confirmed host species. And one important note that I'm curious from kind of a biology of the pathogen is I haven't seen any evidence that palms that grow in like clusters, like the Areca palms, cat palms, things that have those kind of bizarre growth patterns. The palms that seem to be getting hit are the palms that have just a single woody trunk. And I'll touch a little bit about why I think that is in a little bit. Okay, symptoms. Now this is where, as we learn more about the disease, the symptoms are somewhat reliable. If you can see the whole progression, they're reliable, but if you're seeing symptoms, it's too late. The palm's going to be dead. So what basically happens is that in any phytoplasma that attacks a palm, whether it's the A, B, C, D, E, or F strain, you always get the same symptoms, where you have a premature fruit drop, necrosis of the inflorescences, and that's generally useful. But there are some species of palm that have a very temporal pattern to when they're putting out flowers and fruit, like Robolinis, it's once a year. Phoenix dactylifera, I think, also is once a year. So if the palm gets infected during a time of the year when it's not producing these structures, you can't rely on that. But something like a coconut, where it always has fruit and always has an inflorescence emerging, it's a pretty reliable character. And it's one of the characters that sets it apart from fungal infections, because you can get a heavy fungal infection in the canopy and still have some fruit or a healthy inflorescence on one side. So that's a key way to kind of distinguish between the two diseases. After the inflorescence kind of rots off, or the fruit drop, the next stage is that you start to see discoloration of the oldest leaves in the canopy, and that discoloration starts to move further up into the canopy, killing off younger and younger leaves. And this pattern of decline in the canopy is characteristic of the phytoplasma because of what the phytoplasma is doing to the plant. Basically, the symptoms and death of the palm are a physiological response due to stress, because the phytoplasma is invading the vascular network and basically starving the entire plant of nutrients, and it dies. So that discoloration in leaves is a stress response to the phytoplasma, not a toxin or something that the phytoplasma is doing to the leaf tissue itself. And I have some data to show you a little bit later that further supports that, and also highlights why, they call it lethal yellowing, and that's because they saw yellowing in the older leaves of coconut palms. But in LY, it does the same thing. It's the older leaves, it's the premature fruit drop, the necrosis of the inflorescence, and then dying of the older canopy and moving up, affecting younger and younger leaves. And then the final stage is that when the palm dies, the spear leaves collapses, and that's it. So now I'm going to start getting into some of the more applied aspects of sampling and our understanding of the disease. And basically, the best way to really confirm whether it's the phytoplasma is to sample it and test it. And basically, to do that, all you need to do is drill a hole into the trunk of the palm and take out some of the trunk tissue and put it in a Ziploc bag and ship it to my lab, and we do the testing here. And basically, what this is doing is you're drilling into the trunk where there's vascular tissue where the phytoplasma resides, and we do a molecular test where we extract DNA from the sample, and we do a test on it to verify the infection status. Now, it's beneficial to do that for various reasons. If you're trying to do management in an area, either a natural area or a nursery, having those results can help guide your efforts. So it's, you know, having the proper data along with the sample and the sanitation of the equipment is crucial. And we recommend sterilizing the drill bit if you're taking multiple samples and rinsing it off, because if you have a stand of, let's say, 20 cabbage palms and one of them's infected, and you don't clean between sampling the palms, there's no risk in infecting a palm mechanically with the disease. But what happens is if you sample a sick palm, that any residual tissue is going to go into the sample bags for all the other what are healthy palms. When we get those and test it, we get a bunch of false positives, and then we have 9 plants out of 20 rather than 1. And then if your option is to remove the palm, you're going to needlessly destroy 8 plants, hypothetically. So that's why we recommend rinsing and sterilizing. I won't go into explicit detail on the protocol, but I think at the end of my talk, I put my lab's website up there, and I have links to instructions on how to sample, what you need, EDIS documents, submission forms, everything you're going to need to know. But that, just to kind of give you some awareness that we do that testing, and whether it's for management, some people need those types of results for insurance claims. They're starting to regulate movement of plant samples, and I think in the future, testing is going to be a big part of being able to move stuff. We just helped out an organization in Antigua and Barbuda they wanted to import palms from Florida, so I was helping them. I went down and we did a bunch of sampling, and I'm testing the plants for them before they ship out to make sure they meet the standards of being quote unquote healthy. So that's more or less the sampling protocol. So now I'm going to get into and that kind of covers the basic some of the more background information on the disease, the basic biology, how it got here, its distribution, host range, and sampling. So now I'm going to focus more on where we're at, what data we have on management options, and how to slow this thing down. So there's 3 major areas of research we're working on to develop a comprehensive management strategy. Excuse me. The 3 areas that we focus on are aggressive sampling and testing strategies of palms in areas that are at risk or where the disease is. Preventative or the use of antibiotics in palms, and also vector control. So those are the 3 major areas of research. And I'm going to kind of go into detail on each of these. So the first area is we do a lot of applied research here on guiding efforts and decision making on when and if palms should be removed. And basically, from our research, we have found that in this picture here, you can see there are On the left, there's a healthy cabbage palm. Just to the right is early symptoms. To the right of that is moderate. I would say moderate, and then on the end is late. It's at that stage the palm is dead. Now, it's somewhat counterintuitive, but the palm on the right that's dead, that's when people start to get panicky, and then they start ripping out those palms. The reality is, at that stage, the palm is no longer a risk for spreading the disease because there's no green tissue, living tissue for the vector to acquire the phytoplasma. So yeah, a palm that looks like that is an eyesore, but it's not really dangerous for further spread. The dangerous ones are the palm on the left that looks healthy, but it's in the latent period before the symptoms develop, but it still has the phytoplasma. Ironically, it's a trend we're seeing with this the COVID-19. It's when there's no symptoms and nobody's looking at that or thinking there's a disease, that's when the disease is spreading. And I'll show you some data on the OTC here in a moment, but basically a positive test result on a palm is a death sentence. Right now, there's no amount of OTC we're aware of that will cure a palm once it's infected. So, and I've had to write various letters to people explaining in great detail because the landscape service is telling them they need to remove a palm and it looks fine, but it's testing positive. And that's a very difficult thing for people to do. But the logic is that if you don't remove that in the appropriate time, the longer it sits in the environment, the higher probability that the disease is going to spread to more susceptible plants in the area. So basically, when we send out our results, if a plant tests positive, I just tell them remove it immediately, regardless of what it looks like. Because if you don't, it's going to just sit there and be cause further infections. And then once you get to the early symptoms, the palm is still very dangerous because it still has the spear leaf, which is an important factor that I'll discuss here in a moment. But then you can see here, once the spear leaf is collapsed in the third picture and it has that kind of dip in the top, that palm is not really risky for spread because the spear leaf is gone. And I'll go into detail about that here in a moment. So basically, we determined all this, both, the level of phytoplasma in the palm and how to sample it and where to sample it, and looked at different stages of decline. And what we did was we mapped the distribution of the phytoplasma in the palm trunk itself, which helps us answer some basic questions about its biology, but it's also useful from a practical standpoint by giving better recommendations for sampling and testing. And basically, what we did was we created this grid pattern on palm trunks. And the protocol we use in our lab is called quantitative PCR, and it allows us to amplify a specific gene of the phytoplasma. And the software allows us to calculate approximately how many copies of the bacteria are present in the palm. And that way we can estimate where the highest levels are in an individual plant. So basically what we did, we started with this cabbage palm here. And this is the graph that we produced after we analyzed it. So we took well over 200 samples. And basically, the different colors you see on the top graph represent a section of part of the trunk. And then 1 through 33 is how many segments. I'll back up so that's a little clearer. So you can see this kind of pie chart here, and it's a cross section of the trunk. And we divided the trunk into segments labeled 1 through 33, and then each segment had 8 sections around the circumference. And we took a single sample from each of those sections going up the entire length of the tree. And that's how we mapped the distribution of phytoplasma. And the take-home message from this, it's kind of a hot mess in this graph, but it tells us a few things. That when there's symptoms in a cabbage palm, the phytoplasma is fully systemic. It is present in the entire trunk. There's no spot on that trunk that tests negative for the disease. And it also shows that there's an uneven distribution of the phytoplasma around the circumference. So for example, segment 4, section F, the green line, had a lot higher phytoplasma than the other ones. Then when you go to segment 9, section D had a lot more phytoplasma. So you can see it's not the same spot on the trunk every segment that has the highest level of phytoplasma. But basically this tells us that if a palm's symptomatic, you don't have to take a bunch of different samples at different spots. You can get away with just one sample and you're going to get a positive. Now we replicated this with Phoenix sylvestris because this is the most common plant that palm that we get in the for my diagnostic clinic that people send us samples. So we replicated it. We did a early symptomatic, the one on the left, moderate symptoms in the middle, excuse me, and then late symptoms here on the right. And we did the same thing where we divided it into segments and then sampled it sections around the circumference. This is where the data really starts to get interesting. The top graph is the early symptomatic palm, the middle graph is the moderate, and the last graph is the late stage palm. Now, interestingly, when it's early in the disease cycle, in the symptom progression, you have a higher level of phytoplasma lower to the ground. And then as you go further up, you get less and less. You see a similar trend in the moderate symptoms, but as the disease progresses, you start to see more and more phytoplasma increasing higher up into the trunk. Then once you get to late stage, the palm's dead. There's very little metabolic processes, and the phytoplasma is dying off. So you get kind of a loss in phytoplasma titer. But what this tells us is a lot of plant pathogens, when they're injected into a plant, they usually migrate to the roots from where they're injected. And then from the roots, they're pushed systemically through the plant. So in an early symptomatic palm, what has happened is the phytoplasma infects the palm and it's invading the cells for a while. We think about 4 months before symptoms progress or develop. Once symptoms develop, at that point, the phytoplasma has reached the roots and it is starting to replicate at the bottom and push its way up. And it's there that it's starting to block the nutrients. And this, as I mentioned earlier with the symptom progression, when you get this strong stress on a palm, the oldest leaves start to brown up first. And any plant that has one of the, any kind of physical stress usually throws the older leaves first. You see an identical symptoms as lethal bronzing when you see a cabbage palm that's been freshly planted along the highway, because they've cut the roots and they've put it in this new environment. So its stress response is it gets a layer of these bronze-colored leaves that are closer to the ground. So the same thing's happening with lethal bronzing. It's a stress response to the phytoplasma that the leaves bronze up, and then the older ones start to die, and then it moves further up into the canopy. Then as the disease progresses and it makes its way to the spear leaf and the heart tissue, you start to see an increase in titer further up on the trunk, because there's a lot of vascular tissue there, and a lot of the nutrients that the plant is producing are going there because it's the main point of growth and development. So when we get kind of in the middle stage of the disease, you kind of see still most of the pathogen at the base. It dips off a little bit, but then you get an explosion near the top of the trunk because it's right below that apical meristem. Okay, so now I'm going to talk a little bit about the antibiotics and the research we've been doing on that to manage it. One more comment I wanted to make. So to finish that up, that's also why we recommend, even if it's early, it's looking good, or it's just starting to show symptoms, that's why it's the most dangerous, because you can see that it has the highest level of phytoplasma at the base. And if it has invaded the spear leaf and the palm is healthy and still growing at that point, the spear leaf is still desirable for the insect vector to feed on. So that's a key component to this. So while the titer in the trunk at the top might be low, if it's in that starting to invade the spear leaf but hasn't developed symptoms yet, that means the plant is still favorable for the vector, and that's kind of compounding the problem. Okay, so for OTC, there's a label rate where they recommend it for a preventative injection in palms quarterly, basically indefinitely. And all that recommendation is based off research done with lethal yellowing. Now, excuse me, lethal bronzing, it is a different phytoplasma strain. There's no real reason to think it would behave differently in response to antibiotics, but we wanted to be sure. But also because it's infecting new hosts, specifically cabbage palm, we came up with an experiment that we wanted to see what would happen if we injected infected palms. And we also screened it as a preventative measure. So we took healthy palms that tested negative, early symptoms, moderate, and late. And we injected some with an antibiotic, OTC, and then some, our control groups, we left to go natural. So basically here in the cabbage palm on the top, that is the treatment where we injected OTC using the label rate. We saw a significant decrease in symptom progression. So you can see that 3 months after we injected that early symptomatic palm, there's very little symptom progression. The spear leaf was still intact. It was only, I think, another 1 or 2 leaves that had died. But relative to the control, where we did absolutely nothing, 3 months after that first symptom, we noticed the first symptoms and tested. You can see on the right, that palm was completely dead. Then if you stretch it out, the treatment, even 9 months after the OTC was administered, and we did it quarterly, so we kept injecting it per the label rates, it still had some green to the canopy, and the spear leaf was still there. So this is that other palm showing that even at 6 months, or excuse me, 3 months, it was still, it was completely dead. So we saw a significant difference between the treatment and control palms, meaning the OTC was having an effect on the palm, which is encouraging we know that it's effective against the phytoplasma. The problem is the OTC was not curing the palm. It was just slowing down the decline. Now, the issue with using that as a strategy is if you treat it but not good enough, you're just delaying the death, meaning the palm is present in the environment for a longer period of time, increasing the probability of further spread of the pathogen. So this is a case where if you have a palm that tests positive, don't try to inject it with OTC to hang on to it, because you're likely going to create a bigger problem further down the road. It's best to just let it die naturally if you don't want to put up the money to rip it out, or if, the best option obviously is to destroy the plant and get it out of there as quickly as possible. So the encouraging thing is that we have a significant effect of on symptoms progression using label rates of OTC. The interesting thing is when we tested the plants to confirm that they're infected and how much phytoplasma, we couldn't find a difference in phytoplasma titer in the trunk where we were sampling. You can see here that there's no difference between phytoplasma levels, even though we saw a significant difference in symptom progression in cabbage palm. This is pretty simple to explain, that usually when you inject the OTC, it translocates to the canopy, specifically the spear leaf. And basically what we have is the OTC was having an effect on the phytoplasma, but not in the trunk. It was affecting it in the heart tissue, and we didn't have access to that. So we saw a clear effect of the OTC on the phytoplasma in symptoms, and because of the fact that it's not eradicating the infection, as shown here, that you still get positive samples on the OTC and injected palms. We do recommend that if a palm tests positive, don't try to treat it with OTC, remove it immediately. If it's healthy looking and tests negative, then you're going to want to inject it with a preventative injection of OTC. And we kind of couple this to the tree removal program, where if you send in a sample and it tests positive, we recommend, if there's other susceptible hosts, test all of them to make sure you don't have a palm that is in the latent period. So it doesn't, where it doesn't have symptoms, but is still infected and will eventually it will succumb to the disease. So we kind of use these two areas of data together to guide which trees need to be removed from a habitat and which ones should be treated preventatively with antibiotic. Okay, so now I'm going to go and start discussing the vector component of this. When I started, we started doing survey work at the station where the disease was actively spreading. And over the course of a year, we found these 4 species of insect consistently on the palms here at the research center. And the one on the top, Haplaxius crudus, is the species of insect that was determined to be the vector of lethal yellowing. So it was one of our prime suspects from the beginning. These other 3 were present, but they weren't really high targets because none of them are really known to transmit phytoplasma diseases. But what we did was we collected these individuals every week for a year, and we tested them for the presence of the phytoplasma. And basically what we found was that the only species that tested positive of these 4 was Haplaxius crudus. Now, from a scientific standpoint, we have to do transmission assays to definitively prove that that insect can transmit. And we're doing that now. We're working on transmission assays, but the logistics of doing transmission with phytoplasmas and palms, it's a logistical nightmare. The data we obtained from this study, where after a year, every week sampling, and only one species tested positive for the phytoplasma, I'm confident enough that it is the one transmitting it, especially given that it transmitted lethal yellowing, to go ahead and start developing management ideas targeting this species. Because if we wait to, for confirmation that it is vector by transmission, it could be 5-10 years down the road, pretty much. So we're using some indirect evidence of it being a vector, and at this point we're pretty confident that it is. So we're moving forward with developing management programs on this insect. To do this, understanding the vector biology is absolutely crucial. So we're studying the population phenology, basically of the adults, knowing when they have their peak activity during the year and when they're less abundant. That helps guide whatever management or monitoring you're going to be doing, because if it's approaching summer and the population's starting to explode, you need to know that in order to apply the most effective management decisions. Also, we're interested in the distribution of the immatures of this insect, because the nymphs don't feed on the palms. They feed on the roots of grasses around the palms. So it's a very different habitat. So even if you're spraying the canopy with insecticides or using a systemic to control the adults, you're missing the nymphs, and they're just going to keep flooding into the area and continue to cause problems. So the idea is we're developing a system to survey for nymphs in a nursery to determine where they are, so that we can develop a management option to target the nymphs. And we're also looking at the phenology of the nymph population, because the nymphs could be at peak abundance at a different time of year than an adult. So this way it gives us a very flexible and dynamic IPM program that, when the adults are most active, we focus in the palm canopies. When the nymphs are most active, we focus on the grasses where they're developing. So these kind of details are crucial to get ahead of this disease in the nurseries. Also looking at the adult distribution in the nurseries, because there seems to be a feeding preference on what palms they're on. So all these details we really need to understand in order to start controlling spread in the area where I see it being the most common and also the greatest risk to everywhere else. Now, natural stands of cabbage palm way out in pristine habitats probably aren't high at risk, because Haplaxius crudus really likes rather disturbed habitat. So, these tree islands out in the middle of the Everglades or big cypress where there's a lot of cabbage palms, under normal circumstances aren't going to be at risk for the infections. It's the ones that are bordering the nurseries and in the urban environment that get infected as a result of infected palms and insects getting planted nearby. That's, the vector biology plays significantly into where palms are most susceptible and where they're the most dangerous. So the cabbage palms can really get hit when they, not when they're in their natural habitat, but when they're dug up and plopped close together in a disturbed habitat. That's when they become high risk for the infection. So we have some preliminary data on the adult population phenology. This is based on data gathered around the Fort Pierce area, which is where a lot of these palm nurseries are. So that's the most informative. You can see that we tend to see around this time of year is when we really start to see a huge increase in numbers of Haplaxius crudus on our sticky trap. There's two other clusters of graphs to the right of the H. crudus. You don't need to worry about those. It's the one on the left that shows that in the blue bar is September trap captures, and we had a lot more than the other months. And we're still collecting data and trying to make sure that that peak activity is consistent from year to year. That's a crucial element because you know, the effect that climate change has on a lot of these insects can allow for multiple generations per year in some cases. So what historically they may have been restricted by cooler winters where you get a single peak in the summer, it might be possible eventually to get two distinct peaks. So knowing what the year-to-year trend is incredibly important for developing management strategies. And any nursery grower is going to have to continually monitor the populations indefinitely because there are going to be slight variations from year to year naturally, just by natural change in the weather from year to year. But you need to be sure you're catching the population at the right time to do whatever management options you're going to be employing. Right now, we're working with the nursery to do some pesticide trials. And we've done a few treatments so far, and we're doing a few things where we have 3 plots of palms treated with a systemic insecticide, dimethoate, to target the adults in the canopy of the palm. We have 3 plots where we have a foliar spray targeting the grasses where the nymphs are. And then we have plots where we're combining the treatments. And then we have a biocontrol plot to see, you know, Haplaxius crudus is a native species also to Florida. And I've seen natural areas where there's no crudus, but there should be because of the number of palm hosts that are on top of each other, but they've let the plots go. And I suspect that there's a significant amount of biocontrol going on if it's a somewhat intact habitat. So it's a bizarre scenario where you have 2 native species, one of them's killing the other, but the phytoplasma is not a native species. It's from somewhere else. So I've been stalled and slowed down significantly because of the pandemic. So I have months of data sitting in the freezer that needs to be counted and analyzed and updated. And it's just been kind of tough lately to make any progress. But luckily, before everything got really bad, we got some pesticide, some treatments out, and the results were encouraging. I don't know where that slide went. Basically, our preliminary data that we can tell from our initial trap captures is that there's a strong treatment effect for the dimethoate treatment in the palm canopies at 2 weeks, and you can still see the effect at a month. But it's looking like if you want to get, if there's strong disease pressure and you really need to get the population of the vector below the threshold to where the disease can't really take hold, it's looking like treating these plots for, you know, every 2 weeks to a month is going to be the way to go until you get that population under control. But like I said, continual management. So when that population starts to rebound, you want to re-implement these strategies. So that is where we're at with the insecticide trials so far. And we're not doing much in the urban habitats to control crudus, simply because there's so much variability in habitat between, road medians, private households, business parks, public parks, and the crudus is too widespread and patchy that we can't really rely on it. We have to do this in a nursery where we get a very defined plot of a susceptible host where we know crudus is kind of uniformly distributed, more or less. We've also done a few other experiments where we're testing the trap color preference for crudus, and also where in the canopy the insects are most abundant. These were very preliminary experiments done, and unfortunately my postdoc who was working on this left in the middle of it to take another job. So that was somewhat recently, so my world's been thrown into chaos trying to make sense of everything that he had been doing, and his project was the management in the nursery. So that's kind of where I've been focusing on, making sure I'm up to speed with where we're at on that. So at this point, we're shifting heavily into management programs of crudus in the nursery, because that's where I think we're going to have the most bang for our buck in terms of getting ahead of this disease. It's more of a long-term hope that if we can reduce populations and spread in the nurseries, over time that will trickle into fewer infections in the urban environment. Another aspect of control that going forward we're going to be looking for is more cultural control of the nymphs. This is an image of the immature Haplaxius crudus, and as I said, they live in the grass, and we're interested in doing things like weed control, flooding the ditches, removing insects this way. So that's another area we're going to be interested in pursuing in the near future. But we're making progress on understanding the spread and making good progress, I think, on establishing how to control the vector of the disease. We still have a ways to go, and I'm hoping that, so the plan is these management experiments we're doing, we'll have confidence in them by the end of this year, so that next year, basically I'll tell the nursery, this is going to work, we have the data to support it. Start implementing it in your nursery area-wide, and I'll work with them to continue sampling the sticky traps to monitor the effect that the management is having, with the assumption that they're going to be treating their entire palm plot. So we'll put our experiments into practice, I hope, next year, and then track the effect it has on crudus populations, and also try and correlate that with disease incidence in these nurseries. So we're starting to make progress. I just hope that it's not too late. This is my email. That's the website. You can go on to download all the instructions for sampling, submitting samples. I have my EDIS documents. I put all my the publications that we have coming out of the lab that are, some are more basic science, some applied. but they're there for you if you want. And then I have kind of like background information on each of the projects we have going on in the lab. So you can check that out. My phone number if you ever need to reach me, and my address. And usually I am more than happy to host people in the lab if they want to see the process that we go through from sampling to extraction and testing. But excuse me, UF won't allow us to have visitors these days, but hopefully someday soon we'll be able to do that again. So with that, I'd like to thank you for your attention, and I guess we can start doing questions if anybody has any.
Yeah, we do actually have a few questions.
All right.
Eugene Kelly, our Policy and Legislation Chair for the Florida Native Plant Society, asks, is there any evidence of immunity observed among individual cabbage palms?
No. Short and sweet answer is no. Every plant that has tested positive that didn't have symptoms has eventually succumbed to the disease. We haven't found any specimens that test positive and don't get symptoms or die. And then we don't, until we can get a reliable transmission assay and control the introduction of the pathogen to the plant, it's hard to say if some palms are resistant, or whether it's some species are, or if there's individuals in a population that are resistant. We don't know that. I don't think that's the case because it seems like a very low proportion of the vector population carries the phytoplasma. So if there's a susceptible species in an area, the fact that not all the palms get infected, I think, is a result of it just being probability, that because there's so few individuals carrying it, it could be just one or two in that area that are causing the death of 10 palms. Now, if 30% of the population carried it, then you could start to expect to see the entire plot of palms go down. Now, it's possible that some species of palm are resistant to the phytoplasma. Some of those hosts that I documented, specifically Chinese fan palm, I think there's something going on with resistance or immunity to the phytoplasma. I mean, we've documented it in it, but I have evidence that, kind of anecdotal evidence, but at the nursery plot we've been going to, an entire area of hundreds of Phoenix sylvestris went down from lethal bronzing. Right next to that plot is a plot of Chinese fan palms. And it is not, I find it impossible to believe that an infective insect from one of the Phoenix palms didn't feed on the Chinese fan palms in the adjacent plot. Now, when I first started going to the nursery, they told me that they've never had any problems on Chinese fan palms in that plot. And at first I was like, oh, maybe they're not being upfront. But after years of working there, I've never seen any of those palms die. It seems like the Chinese fan palm, at least, looks like there's some degree of resistance to it. But until, like I said, until we get a good transmission assay, it's impossible to say.
Thank you. That was a very thorough answer. M. Stroud asks, is there any reason why Volusia County seems to not have any cases of lethal bronzing?
Which county?
Volusia.
Sorry, my Florida geography is not phenomenal. Where's Volusia?
It's south of Flagler, Daytona Beach, north of the Space Coast.
So it's there. I know it's there, but we have not received samples from there. Now, and this is one of the challenges to this work and getting samples is we rely heavily on people sampling and sending stuff in. Now, a lot of those counties that haven't, I showed you on the map, it's not that they're definitively free of the phytoplasma. It's that we just haven't gotten a sample from it yet, or somebody hasn't noticed it yet. So recently I drove up, uh, um, I did a road trip because I was trying to determine the northern limit of Haplaxius crudus, where it stopped um, persisting in the natural environment. And I was collecting along the way, and um, you I can see LB infected palms all the way up to Jacksonville. There are spots where it's not as bad, but um, they are there, and I didn't have uh, my students had the palm collecting gear, and we're off doing another experiment, and I just had my bug collecting equipment, but I know it's in that area. It's not that it's not. It's just we haven't gotten a positive sample from that area yet. Yes, there are plenty. Pretty much any in a semi-natural habitat, uh, there's lots of spiders, and one that I'm kind of curious about. It actually, it's rather annoying when I'm trying to collect crudus. It's a jumping spider that's about the same size, it's narrow, and it's the same color. So I don't know if it's a mimic of crudus, so it can get in closer, but I always find those on cabbage palms. So I know just about any spider in that habitat is going to feed on crudus. Um, I'm I suspect that there are parasitoids, wasps out there that are attacking them, but I don't know that for sure. But at least the generalist predators like assassin bugs, um, dragonflies, those types of things that will eat anything they can catch. If it's a habitat that's conducive for those natural enemies, then certainly they do a significant amount of control.
Jono Miller asks, has anyone been looking at palm hammock impacts adjacent to sod farms?
Well, that's a good question. Um, Not yet. That's kind of on my radar for looking at closer because the sod is, I do think, going to be an issue because the outbreak that happened here in Fort Lauderdale was in 2014, and before I got here, so I cannot be blamed for it. But basically, what happened was they had done some construction here on the center, and they had ripped up all of the landscaping and the turf grass. Then they came in and had to resod it, and they brought in big pallets of St. Augustine grass, which is a well-known host of the nymphs of Haplaxius crudus. So they brought in all this sod, and then 4 months later, lethal bronzing shows up at our front doorstep. So what I suspect is that an insect carrying the phytoplasma was a female who had just laid eggs, and then she was moving back to the palm canopy to feed. Um, so the I do suspect that palms near sod farms are going to be more at risk, um, but that's something that I haven't pursued too much yet, um, because I know when I start, shaking my finger at the sod, the St. Augustine grass industry, I'm going to have problems, and I want to wait till I have tenure for that. So,
Jono also asks if you're planning an in person or a Zoom lethal bronzing workshop for this fall.
um, I really want to, and I'm trying hard to make that happen. I will admit I have been neglecting that aspect as of late, uh, mainly because my main research project that I was putting a lot of time and money in was with my postdoc on these nursery trials. And basically, at the peak, he left, and I have had to be the one doing, uh, going out and doing the field work, the data collection, and I have to go out every other week. I have to count the traps. Uh, I'm and now I've been doing a lot of paperwork on my grants to get extensions because we haven't been able to meet the deliverables because nobody's allowed in the lab to do work. So it has um, thrown off everything. So I've had to put my energy into uh, basically the main part of the research program, and unfortunately, it has taken away from my energy and availability to do some of the workshops. But every year I've done a lethal bronzing workshop here at the center, and I was intending to do one this year, but it's not going to happen. UF won't even allow me. But basically, I'm still entertaining the idea of doing a holding it virtually, but I haven't decided yet because one of the big things that's really beneficial about that workshop is having people come out, see the disease in action here at the center, see what we're doing, and I'm not sure about that workshop. I will be doing. I know for sure before the end of the year I'll host a smaller one at least on um, maybe just a live one like sampling. I'll have my student follow me with my phone, some of the more important aspects of it. But I'm still not 100% sure about the day-long workshop. If I do, you can if I do decide to do it, you can check back on the Fort Lauderdale website. Is when I'll basically advertise it there, and you'll be able to find out the date. Or you can just email me and bug me over the next month or so, and I'll let you know if what I've decided.
Thank you. I'm sure there would be a lot of interest for that. Eugene asks, what can FNPS do to assist in controlling lethal bronzing? Education campaign? Funding needs? Collecting cores?
Well, funding is always a critical component to this, and right. So we've in the last year or so we've done fairly well. Um, the ISA Florida chapter has been very supportive. They've gotten us a lot of money. The National Horticulture Foundation has funded us. I got some big farm bill money from the USDA. So we've gotten some funding. So right now we're doing okay, but given the state of things, I don't know what kind of funding is going to be available in the years to come. So I have adopted the mentality even before the pandemic that in terms of research, especially on this, there's no such thing as too much money. The more we have, I can bring on more students, more staff, and we can move this along faster. So funding obviously is always a help and a beneficial one. The kind of the public education I think is going to be critical. And there could be minor things with homeowners and HOAs where they're potentially vigilant if they're buying sod from a landscape company or from Home Depot, know that it could come with a risk. I had contemplated at one point, once we get more involved in the sod research, some experiments I wanted to do would be to take like a piece of sod and put a bunch of Haplaxius crudus nymphs in it, let them settle in for a day or two, and then submerge it in water and see how long it takes to recover 100% of those nymphs. So that basically, if there's a shipment of sod going out, we can recommend that the sod be flooded for X amount of time, or a public awareness campaign with Home Depot, put up like a warning sign next to the sod saying, oh, this could be carrying this bug. If you are going to buy sod, put it in a bucket at home for an hour or two with water to get rid of all the bugs, and something like that. I think going forward may not be enough to stop the disease, but every little bit counts if everybody chips in and contributes. So yeah, I think always, like with many things, funding and public awareness and participation are going to be huge helps in stopping this. And the other thing from my research end that I'm starting to realize I would be beneficial is getting in contact with as many nurseries that grow palms who are willing to allow me to monitor for Haplaxius crudus and would be willing to treat their palms with systemic insecticides and allow me to monitor the effect of it. Because the more data I get on that, the better. And the more data I have, the better recommendations and advice I can give the people towards managing. So that's another key component that if anybody listening has a nursery or knows somebody who would be willing to invest in treating their palms and letting me kind of shadow to look at the effect. I've gotten to the point where I've contemplated getting my pesticide applicator license, buying an ATV and applying the insecticides myself here on the station if I couldn't get nursery collaboration. But the nurseries are really crucial. It's a different habitat than here at the center, so that's something I really need help on, is getting nursery collaboration to where they can where I can work with them and monitor their application of or whatever they're going to do to control the bug and assess the effect. In a perfect world, like, if a nursery doesn't want to apply insecticides and they just want to let their weeds grow, I can use that as my biocontrol plot. I can collect all the predators, parasitoids, and monitor crudus. If it's a nursery who is, set up for pesticide and they're fine with doing that, then I can use that as my treatment plot, where I say, okay, I want you to treat, once a month, and I'll come out and collect the sticky traps the morning before you treat. Just let me know. And some kind of a collaborative research effort between me and the nurseries is really what I'm working hard towards developing in the next couple years.
Awesome. Diane Goldberg asks, what are the grasses that host the nymphs?
Well, it'd probably take me less time to list the grasses that aren't host for the plant, the Haplaxius crudus. They found in one study in Brazil that there are over 70 species of grass that host Haplaxius crudus. So I'm willing to bet if it's a grass, crudus will feed on it. Now, obviously, I think there are some species that are more preferential, but we don't know exactly which ones. In Florida, St. Augustine grass in urban environments is the preferred grass. In the nurseries, it's a rather large grass. We're trying to identify actually what it is, but it's a rather large clump of grass, and they seem to like that a lot. So, but like I said, I think any grass is going to be fine for them.
Is there any breakdown in nativity status, like they prefer more native grasses or bunch grasses or non-native grasses?
No, I don't know. I don't know. Once the other thing that we're trying to do is get a good colony started, um, so that we can have access to as many individuals and then do experiments in terms of um, egg laying preference. So basically, we have a couple of different grasses in an arena, and then we put in some mated females and uh, see where they go.
Do palm trees, do any of these palm trees that are infected with disease or any that aren't have any chemical or physical defenses against any of these nymphs or any of these plant hoppers?
No, and that's one of the problems I think with palms is they don't have uh, really any good defense mechanisms like dicots and woody plants. They don't have a really good healing system or immune response. Um, but honestly, I don't know for sure, and I don't know if anybody's looked at uh, looked at that in terms of phytoplasma. I know I haven't, but I'm not sure if they did that with regard to lethal yellowing in coconut palms. But from my end, I haven't. I haven't done anything, and I'm not aware of anybody doing anything on that. The closest to that we're doing is maybe host vector feeding preference with, or looking at if an infected palm is more desirable to the vector um, versus a healthy palm. So that's another, to see if it's releasing a volatile chemical that they're detecting or not. So that's the closest to that we're investigating right now.
Thank you. There is one last question from Diane. She says, "Aren't you worried about systemic insecticides killing pollinators?"
What's that?
She says, "Aren't you worried about systemic insecticides killing pollinators?"
Um, it's a concern. The whole idea of systemics is that it reduces exposure to the pollinators. It's not like they're getting blasted with the insecticides. Um, that being said, I'm kind of in the boat of that. The issues with pollinators for me is more of a combination of a lot of factors. And yes, insecticides to some degree can have an effect on them. There will be some that will die because of this, and it's unfortunate for sure. But the impact at this point, the impact of the disease on a native palm is it's urgent. So we push the systemic to try and reduce the impact of the insecticide, but there will be casualties. It's unavoidable, but we're trying to. What I see is, and I'm not a pollinator ecologist or or researcher. I'm work on this disease, so naturally I'm trying to solve this problem, and then I understand that there will be some impact on the pollinators, but we want to try and get this disease under control before it has too much of an impact on both native or naturally occurring palms on the edge of natural habitats and in the nurseries. Then once we get a handle on the disease, then you don't have to use insecticides as much. We'd just be part of a monitoring program that if things start to flare up, you use your treatments at the right time of the year. Like I mentioned, with the phenology of the adults of Haplaxius crudus, knowing when they're at their peak activity, that's probably when most of the transmission is occurring. So instead of just blanket insecticides throughout the year, if we know when the highest risk of transmission is because of high numbers of vector, we can limit our time to treating palms, for a single month out of the year rather than 12 months. So there are factors that I will admit aren't my priority in thought process of protecting pollinators, but the logistics of how we're doing some of this will inadvertently result in less of an impact on the pollinator. So I'll admit that it's not that I don't care about the pollinators, but they're important, but it's not at the forefront of my concern in this research.
Right. And one final word from Eugene, and I echo his sentiments. Please reach out to FNPS if or when you need the kind of assistance or advocacy we might be able to provide. We're in your corner, and our members are very concerned about any damage to our state tree, our native Sabal Palm.
Does he have a contact that I
Yes.
can I just reach out to him directly, or?
Yes, he would be the best contact. I'll email you.
Okay.
I'll send an email introduction right after we get done.
I think we've corresponded in the past, so I'd have to dig, but just to be safe, if you can forward it to me.
Yeah, of course. Well, I really appreciate you coming on and presenting to us.
Well, I really appreciate you having me, and thank you everybody for tuning in. And sorry we couldn't do this in person. Hopefully next time we can do it in person. I always feel more valuable. But
yeah, we
we're
really
adapting to an insane new world, I guess.
Yeah. We're doing what we can.
Yeah.
So thanks everyone for coming to the Florida Native Plant Society Lunch and Learn for August 14th, Dr. Bahder, and I hope everyone has a wonderful weekend.
Thanks.